it's now my pleasure to introduce our second speaker. Ms. Ifthiha Mohideen is a graduate student at Concordia University. She is a PhD candidate in the laboratory of Dr. David Kwan at Concordia, and her doctoral research focuses on the function and engineering of enzymes involved in the glycosylation of natural products. In particular, she's interested in developing a method for assaying glycosyltransferases in a high throughput fashion and engineering novel in vitro enzymatic pathways for the anti-cancer anthracycline deoxysugars. Before joining Concordia, Ms. Mohideen got her Bachelor's of Science with honors in plant biotechnology from the University of Colombo in Sri Lanka in 2014. And with that, I'm looking forward to hearing her talk.
Thank you, GlycoNet, for having me. So first of all, I would like to give an overview of my PhD thesis project. So I work with glycosyltransferases that are involved in the glycosylation of small molecule natural products. And in my thesis project, I'm interested in developing a high throughput screening method for these enzymes, and also to use this assay for combinatorial screening with the aim of obtaining novel glycosides. As I have shown in this diagram, and as you all know, glycosylation requires two substrates. We need a sugar donor and a sugar accepting molecule. I'm interested in vitro enzymatic synthesis of certain nucleotide activated sugar donors, and also to engineer a glycosyltransferase enzyme towards a desired phenotype. So today, a major portion of my talk will be on enzymatic synthesis of these TDP sugar donors, and also I will talk a bit on engineering a glycosyltransferase enzymes. So small molecule natural products are produced by different organisms, including bacteria, fungi, lichens, plants, and insects. These structurally diverse small organic molecules are synthesized through secondary metabolic pathways. Considering the importance of these natural products, in 2005, Schreiber suggested that these natural products can be included in the central dogma, and I thought it's interesting. Many small molecule natural products are decorated with one or more sugar moieties, and they have a great effect on their biological activity. And we are interested in these glycosylated small molecules because they show therapeutic importance. Examples include montbretin, a potent antidiabetic, antibiotics such as erythromycin and vancomycin, and anti-cancer drugs such as Daunorubicin and its derivatives. As you all can see, these molecules are highly diverse in structure and biological activity. And out of these molecules, we are interested in anthracyclines. Anthracyclines are a class of small molecule glycosylated products, and in nature, these are produced by Streptomyces species. Some of the clinically important anthracyclines include Daunorubicin and Doxorubicin. Daunorubicin is used for treating acute lymphoblastic leukemia and myeloblastic leukemia, whereas Doxorubicin, it shows broad spectrum anti-cancer activity. However, prolonged administration of these anti-cancer drugs results in dose-dependent cardiotoxicity. The Doxorubicin analog Epirubicin is a high-value anti-cancer drug, and Epirubicin has gained an attention because it shows similar anti-tumor properties as Doxorubicin, but it has lower cardiotoxicity. So if we look at the Doxorubicin Daunorubicin biosynthetic gene cluster, it is around 40 kilobases and it has 37 open reading frames. This includes genes encoding for the polyketide A glycan, structural genes encoding for the deoxysugar, genes encoding for glycosylation, post-modification, and genes involved in regulation and resistance. So we can divide the Doxorubicin Daunorubicin biosynthetic pathway into 3 steps: biosynthesis of the A glycan, biosynthesis of the sugar donor, and glycosylation and post-modifications. Starting from one propionyl-CoA unit and 9 malonyl-CoA extender units, first the rhodamycinone A glycan is synthesized, and then the sugar donor for doxorubicin and daunorubicin is TDP-L-daunosamine. So the reaction begins from glucose 1-phosphate, and sequential action of these two enzymes will produce this key intermediate, and that is TDP-4-keto-6-deoxy-D-glucose. Then the pathway undergoes further dehydration and aminotransferase reaction, epimerization, and a ketoreductase reaction to produce TDP-L-daunosamine. So once there is the sugar donor and the acceptor molecule, a glycosyltransferase enzyme, and in this case it is DNRS, will catalyze this reaction to produce this glycoside. And then a few other enzymatic reactions will lead to the production of Daunorubicin, and finally this is converted to the Doxorubicin and anthracycline. So let's have a closer look at the structures of these two anthracyclines. So if you look closely, you can see the main difference between these two is the orientation of this C4 hydroxyl group, which I have shown in red. Despite the clinical importance of epirubicin, it is conventionally produced semi-synthetically through several organic synthetic steps. However, if we want to produce epirubicin enzymatically, we need three components. We need an enzyme to catalyze the glycosylation, we need the A glycan, which is rhodamycinone, and we need a sugar donor, and that is TDP-L-acosamine with an equatorial hydroxyl group at C4 position. However, a natural biosynthetic pathway for this sugar donor has not been described, and currently it is chemically produced. As an alternative to chemical synthesis, attempts have been made to produce epirubicin and its sugar precursor in vivo. This is possible because many glycosyltransferases and sugar biosynthetic enzymes are promiscuous. Because of the substrate flexibility of these enzymes, in this paper, they were able to produce epirubicin in an engineered Streptomyces species. So they constructed the epirubicin pathway based on the biosynthetic pathway of Doxorubicin and Daunorubicin. And you can see in this reaction, they used a stereospecific ketoreductase to produce TDP-L-acosamine. So they fed rhodamycinone exogenously, and then they used this glycosyltransferase enzyme to produce epirubicin. And they were able to get 0.5 milligrams per liter, which is a low yield. Though they have synthesized this sugar precursor in vivo, they did not isolate it. So in our lab, we are interested in vitro enzymatic synthesis of TDP sugars, and I'm specifically interested in synthesizing TDP-L-daunosamine, TDP-L-acosamine, and some other intermediates. Having pure sugar donors and its intermediates is important because we can use them to characterize and co-crystallize some of the enzymes, and also these could serve as precursors for some other highly modified sugar donors. Towards this objective, first, using this paper with slight modifications, we have synthesized and purified compound 1, or TDP-4-keto-6-deoxy-D-glucose. I have shown the mass spec data for compound 1, and this is compound 1, and this is the hydrated form of compound 1, and I was able to get a yield of 82%. Next, starting from compound 1, we have proposed a one-pot, two-step reaction for the production of TDP-L-daunosamine and TDP-L-acosamine in vitro. So the reaction is initiated by adding these two enzymes. In this case, EVAA will dehydrate compound 1, and then DNMJ will add an amino group to produce compound 3. In the second step, we add an epimerase enzyme, and a ketoreductase. So the choice of the ketoreductase depends on the final product that we want. If we want TDP-daunosamine with an axial hydroxyl group, it will be DNMV. And if it is TDP-acosamine, it is ABEP4. So using our proposed pathway, I synthesized and purified an intermediate that is compound 3. And then I was able to get a best yield of 43%. Also, it should be noted that same as compound 1, compound 3 also exists in a hydrated form. Next, using our proposed 1-part 2-step pathway, I have synthesized TDP-daunosamine, and I have shown some mass spec data. So this is compound 1 in the absence of all 4 enzymes. This is compound 3 in the presence of first 2 enzymes. And this is TDP-daunosamine in the presence of all 4 enzymes. So to conclude, we have proposed an engineered in vitro enzymatic pathway for the production of TDP-daunosamine and TDP-acosamine. And I have synthesized and purified compound 1 and compound 3, and we have sent some of them to Dr. Rongshi at University of Laval for the co-crystallization of some of the enzymes in the pathway. I have optimized the conditions for the production of TDP-daunosamine, and currently I'm working on the ketoreductase that catalyzes this step. And next I will be scaling up and purifying these 2 sugar donors, and we will also characterize some of the uncharacterized enzymes in the pathway. So let's move back to epirubicin production. If you recall in the paper that I showed you previously, they didn't get a good yield for epirubicin. And one possible reason could be poor glycosylation of these epirubicin precursors by this glycosyltransferase enzyme, because the native substrates of this enzyme is not same as this. So if we can increase the substrate specificity of this particular enzyme, we can increase the production, epirubicin production yield. Towards this, we are planning to engineer a glycosyltransferase enzyme via directed evolution. So we will start from AKNS as our target gene, and then we will construct a mutant library. Following transformation and protein expression, we will screen the library. So for the screening, we need the aglycan, which is rhodamycinone. We need the sugar acceptor, and I will be synthesizing this in the lab. So rhodamycinone, it is commercially available. And then we need a high throughput assay for glycosyltransferases. So I have already developed and I have validated a high throughput screening method for glycosyltransferases, and it's simple, it's easy to perform, and it's reliable. But in today's talk, I won't be talking about it. So following screening, once we get positive clones, we will do further rounds of directed evolution until we get a glycosyltransferase enzyme that can efficiently catalyze this glycosylation reaction of epirubicin precursors. Last but not least, I would like to thank my supervisor, Dr. David Kwan, and our lab members, and also all the funding agencies. Thank you.